Welcome to Chapter 18, the Cardiovascular System, Part 2 of 2. In Part 1, we studied the anatomy of the heart. We saw where the heart was located in the chest, and then went on to look at the protective coverings surrounding the heart. We looked at the external and internal anatomy of the heart, and ended with the flow of blood throughout the heart and coronary circulation. In Part 2, we will look at the microscopic anatomy of cardiac muscle tissue, the intrinsic conduction system, and we will finish up with the electrocardiogram. Cardiac muscle tissue is a type of muscle tissue, and we learned in Chapter 4 that muscle tissue is one of the 4 primary tissue types in the human body. Cardiac muscle tissue is primarily composed of cardiac muscle cells, also known as myocytes. These muscle cells are going to be striated, kind of like a skeletal muscle cell. They're fairly short, they branch, they are uninucleated, meaning they have one nucleus, but they have many mitochondria. Remember, a mitochondria is the powerhouse of the cell. Mitochondria utilize oxygen to produce energy in the form of ATP. Similar to skeletal muscle cells, cardiac muscle cells also have sarcomeres. Remember the A band, the I band, the Z disc? Cardiac muscle cells also have T-tubules, a sarcoplasmic reticulum, but no triads. Something that's extremely special about cardiac muscle cells are these intercalated discs. And these are found where we join cardiac muscle cells together. An intercalated disc has 2 components to it. The first is a desmosome. Think of this as the glue that holds neighboring cells together. It prevents these cells from coming apart from each other. And the other part of an intercalated disc is a gap junction. This is an actual space which will allow for the passage of ions like calcium to move from cell to cell. Overall, the intercalated disc is what allows the heart to be a functional organ or a functional tissue. When the heart contracts, it's a single coordinated unit, not multiple. This diagram is showing you a microscopic view of cardiac muscle tissue. So in the image on the left, you are able to see that cardiac muscle cells are also striated. They're fairly short and they branch. You should identify the nucleus or that dark stained structure. But most importantly, you should identify the intercalated discs. And they look like dark lines that are perpendicular to the direction of the muscle cells. Remember, an intercalated disc includes 2 components. The first was the desmosome. This is the glue that holds neighboring cells together. And the second component was a gap junction, or a space, which allows for the passage of ions between cardiac myocytes. We have briefly compared and contrasted cardiac muscle and skeletal muscle before, but now let's look at some details. For the most part, cardiac muscle tissue and skeletal muscle are similar in that they contract the same way. Muscle contraction in both types of tissue comes after an action potential. Remember that electrical impulse that is there to excite the muscle cell. From there, we still have depolarization. Remember that wave of impulse that's going to dive down the T-tubules and stimulate the release of calcium from the sarcoplasmic reticulum. From there, we still have troponin, we have tropomyosin, and we see the sliding filaments, actin and myosin. despite the similarities between these two types of muscle, they also have their obvious differences. In addition to being located in different parts of the body, some cardiac muscle cells are actually able to excite themselves, or they are able to generate an action potential without an external input. But aside from that, we also have two kinds of cardiac muscle cells, or cardiac myocytes. They are known as pacemaker cells and contractile cells. Pacemaker cells make up 1% of these cardiac myocytes. Pacemaker cells are also known as non-contractile cells, cardiac conducting cells, or autorhythmic cells. They are going to generate an action potential without any external input. Therefore, they are self-excitable or capable of generating a rhythm automatically. The other 99% of cardiac muscle cells will be the contractile cells. And these are the cells that we usually think of when we think of a muscle cell. These are the cells that will shorten to generate force or contract. Even though these non-contractile cells or pacemaker cells only make up the 1% of cardiac myocytes, they are often found in clusters or nodes or fibers, and they create this system in the heart known as the intrinsic cardiac conduction system. That name of the system tells you what it does. It is a system that can conduct an electrical impulse intrinsically or without any external input. These cells or this system is responsible for creating and spreading or propagating these electrical impulses throughout the heart. The rate of this system, or the rate of your heartbeat, is set by these cells. It is intrinsic. However, we usually have an extrinsic or an external source affecting it. This external source is known as the autonomic nervous system. In short, the autonomic nervous system controls processes that occur automatically or subconsciously, like digestion, breathing, salivation, or sleeping. These nodes and fibers containing the pacemaker cells aren't just randomly placed throughout the heart. We like to organize them throughout this system, and you have to know the organization and the order in which they occur. Listed 1 through 5 includes the structures, nodes, or fibers in which the excitation moves throughout, or the sequence. So the very first thing that we have is known as the sinoatrial node, which you could simply call the SA node. The SA node is found in the roof of the right atrium and is often known as the pacemaker of the heart. Since this is the first structure in the sequence or in the system, it's setting the pace or the heart rate. Usually it's an inherent rate of 100 beats per minute. Now, if you were to take your heart rate right now, it might be 70, it could be 60. That is because we do have external sources affecting our heart rate, but we don't need those external sources. After the SA node depolarizes or fires, that impulse spreads throughout both the right atrium and the left atrium until it hits the next structure in place, the atrioventricular node, or the AV node. Once the impulse hits the AV node, we pause. And we pause because we need to allow for the atria to fully contract to ensure that all of the blood has been pushed out of them and into the ventricles. After we have a brief pause at the AV node, we then see the AV bundle, or the bundle of His. Eventually, the AV bundle will split into a left and right bundle branch that can be seen in the interventricular septum. The left and right bundle branches will course towards the apex of the heart. Remember, it points towards your left hip. After those bundle branches hit the apex, they swing upward in the ventricular walls as the Purkinje fibers. Again, this is the order in which we see excitation or depolarization. Once these cells fire, their job is to stimulate the surrounding cells, which are the contractile cells. These cells fire or depolarize, and then we see the contraction of the heart. If you're having trouble picturing this, here we have an anterior view of the heart. Be sure to orient yourself anytime you see an image of the heart. First, identify the SA node found in the roof of the right atrium. The SA node is also known as the pacemaker of the heart because it is the first cluster of cells in the sequence or in the system. Once the SA node fires, that depolarization spreads throughout both the left and the right atria, and then it hits the AV node in the floor of the right atrium. After the impulse hits the AV node, there is a pause. There's a pause to allow for both the left and the right atria to fully contract. After the pause, the impulse is carried throughout the AV bundle. The AV bundle will then branch into a left and right bundle branch. Once the bundle branches hit the apex of the heart, they then swing up into the ventricular walls as the Purkinje fibers. Again, this system only accounts for 1% of cardiac muscle cells. When these non-contractile or pacemaker cells fire, they stimulate the surrounding contractile muscle cells, and then we see the heart beat. So when the heart beats, it's atria first, then ventricles. Atria first, and then ventricles. As previously mentioned, these non-contractile or pacemaker cells are able to generate electrical impulses without any external input. That is why it is called the intrinsic conduction system. However, it's very rare that our heart rate is 100 beats per minute when we are simply sitting down, which is where we begin to discuss the autonomic nervous system. The autonomic nervous system is responsible for the fight or flight response, but also for allowing you to rest and digest. The autonomic nervous system is located in the brainstem and contains two centers, which will innervate or control the heart or the heart muscle. The first center is known as the cardioacceleratory center. This center, as its name suggests, is going to accelerate the heart rate. This center will stimulate the SA node and the AV node, amongst other structures. But the heart's response to that center is to increase the heart rate and the force of contraction. This is a normal response during exercise. And on the opposite side, we have the cardioinhibitory center. As its name suggests, it's going to dial down or inhibit the SA and the AV nodes. In response to this center becoming activated, we see a decrease in the heart rate and no effect on the force of contraction. This is a normal response as you prepare to go to sleep at night. Although we are not learning about the nervous system right now, we do have to address the autonomic nervous system. Now you see a bunch of green and purple lines moving about, but all you need to know is that it is possible to alter the heart rate extrinsically. And the autonomic nervous system does that with its two centers. One center will stimulate the heart, and we will see an increase in heart rate and force of contraction. The other center will inhibit the heart, we will see a decrease in heart rate. The last topic of this lecture will be electrocardiography. If you break apart that term, you get electrical events or electricity, the heart, and an observation or a measurement. Electrocardiography is the observation or measurement of the electrical activity in the heart. On the screen here, you are looking at an electrocardiogram or an ECG. These are performed in the clinical setting. They usually involve sticking electrodes on the abdomen and thoracic region of a patient. And what happens is those electrodes pick up the electrical currents that are produced by the heart. Basically, they pick up the activity of those pacemaker cells. The next thing we will do is look at the graphic observation and label the different parts of it. Then we will take it a step further and actually figure out what is going on in the heart to create that observation or to create that measurement on the paper. When studying an ECG, you should identify 5 main waves: the P wave, the Q wave, the R wave, the S wave, and the T wave. Oftentimes we collect the QRS waves to form just simply the QRS complex. Now, not only do you need to be able to identify these waves on a reading, you need to understand what is occurring in the heart to cause that wave to appear on the graph. So the P wave is representative of the depolarization of the SA node. So what happens is the SA node fires. It generates an electrical impulse and spreads throughout the left and right atria. In response to that depolarization, we then see the atria contract. Next, we have the QRS complex, which is easy to identify because it's a very large upward spike on the graph. The QRS complex occurs when we have ventricular depolarization and atrial repolarization. Ventricular depolarization is that electrical impulse that will stimulate the surrounding contractile cells. But while that is happening, we also have the atria resetting or getting ready to have another impulse. And the last wave seen on a reading is the T wave. Usually appears to be a small hill, but the T wave represents ventricular repolarization. The T wave appears when the ventricles are returning to baseline or resetting themselves and preparing for another impulse. In addition to these 5 waves, we also have intervals and segments. And these also indicate certain events going on in the heart. These intervals and segments can often be used to diagnose cardiomyopathies or even heart attacks. In between the P wave and the R wave is the PR interval. And you can actually go in and measure the time of the PR interval. That time tells you the time between atrial excitation up until ventricular excitation. The ST segment, so in between the S wave and the T wave now, that is where we see the entire muscle in the ventricles excited or depolarized. And lastly, the QT interval between the Q wave and the T wave is the time from the onset of ventricular depolarization up until the completion of ventricular repolarization. As previously mentioned, not only do you have to be able to identify the waves on the ECG, you have to understand what is going on in the heart electrically to cause those waves to appear. So again, you have to be able to identify the P wave, the Q wave, the R wave, the S wave, and the T wave. You also need to be able to identify the QRS complex, the PR interval, the ST segment, and the QT interval. This graph on the screen represents one cardiac cycle. This graph represents the atria contracting and resetting, the ventricles contracting and resetting. On this diagram here, we are comparing the heart, or the events in the heart, to the wave, or to the ECG. Yellow represents depolarization. It is excited. Red represents repolarization. It is returning back to baseline. At the very top, remember the P wave represents the firing of the SA node and the depolarization of the atria. In yellow, the heart, it shows you the area that is depolarizing. Now, that line in between the P wave and the Q wave, that flat line represents the pause at the AV node. The pause at the AV node is important because if you look at the heart, both atria are fully yellow. The pause allows for both atria to fully contract. Next, we have the QRS complex. The QRS complex is the depolarization of the ventricles. Then we have the flat line in between the S wave and the T wave, or the ST segment. That represents full ventricular depolarization. The T wave now, you can see that the apex is beginning to repolarize, and it works its way up to fully repolarize both the left and the right ventricle. Again, this is one cardiac cycle. We will finish up this lecture by defining two terms, systole and diastole. Systole refers to heart contraction. During systole, there's usually a high pressure detected in the heart. Diastole is the relaxation of the heart muscle, or the filling of the heart with blood. Both the atria and the ventricles are capable of both. During atrial or ventricular systole, the heart muscle in those chambers is contracting to force blood elsewhere. During atrial or ventricular diastole, those chambers are relaxed and they are allowed to fill with blood. This was chapter 18, part 2. We learned more about the microscopic anatomy of cardiac muscle tissue and cardiac muscle cells. From there, we moved into the intrinsic conduction system. Make sure you know the sequence or the order of structures in which an impulse will pass through. And then we finished up with the electrocardiogram. Make sure you know the waves and what those waves represent in the heart.